Regulation of translation. The regulation of translation refers to the control mechanisms that determine the rate and efficiency of protein synthesis from mRNA molecules. It plays a crucial role in the regulation of gene expression and allows cells to respond to various internal and external signals. Here are some key aspects of translation regulation. First is translational initiation. Translational initiation is the rate-limiting step in protein synthesis and a major target for translational regulation. It involves the binding of the small ribosomal subunit, along with initiation factors, to the mRNA molecule, followed by the recruitment of the large ribosomal subunit and the start codon recognition. Regulation of translational initiation can occur through various mechanisms, such as 5-cap and poly A tail. The presence of a 5-mRNA cap and a poly A tail enhances translational efficiency. Translational repressors can bind to these elements and inhibit initiation, while translation activators can enhance initiation by interacting with initiation factors. Regulatory sequences. mRNA molecules can contain regulatory sequences within their 5 or 3 untranslated regions, UTRs, that can influence translational initiation. These sequences can interact with specific RNA-binding proteins or microRNAs to promote or inhibit translation initiation. EIF2 and EIF4E. Initiation factors such as EIF2 and EIF4E are key regulators of translational initiation. Phosphorylation of EIF2 can inhibit global translation initiation under various stress conditions, while changes in the availability or activity of EIF4E can modulate the translation of specific mRNAs. Second is translational elongation. Once initiation is completed, the ribosome moves along the mRNA molecule and synthesizes the protein through a process called translational elongation. While elongation is generally considered a constitutive process, some factors can modulate its rate and efficiency. First factor is elongation factors. Elongation factors, such as EEF1A and EEF2, facilitate the movement of the ribosome along the mRNA and the incorporation of amino acids into the growing polypeptide chain. Modulation of the activity or availability of these factors can affect the speed and accuracy of elongation. Another factor is ribosome pausing. Certain sequences or structures within the mRNA can cause ribosome pausing during elongation. This can be regulated by RNA-binding proteins, small regulatory RNAs, or specific codon sequences. Ribosome pausing can regulate the translation of specific mRNAs or contribute to quality control mechanisms. Third is translational termination. Translational termination occurs when the ribosome reaches a stop codon on the mRNA, leading to the release of the newly synthesized polypeptide chain. Termination is generally a highly efficient process. However, it can be influenced by factors such as ribosome stalling or the presence of regulatory elements within the mRNA. Fourth is regulatory factors and RNA-binding proteins. Regulatory factors, including RNA-binding proteins, can interact with specific mRNA molecules to modulate their translation. These factors can bind to specific regulatory sequences within the mRNA, such as upstream open reading frames EOFs or internal ribosome entry site SIAs, and influence translation initiation or elongation. They can either promote or inhibit translation based on the specific interactions with the mRNA and the translation machinery. Number 5 is small regulatory RNAs. Small regulatory RNAs, such as microRNAs, mRNAs, and small interfering RNAs, siRNAs, can regulate translation by binding to specific mRNA molecules and inhibiting their translation. mRNAs, in particular, recognize complementary sequences within the mRNA, and promote mRNA degradation or translational repression. The last one is cellular signaling and translational control. Signaling cellular signaling pathways, like the mTOR pathway, impact translation control in eukaryotes. By modulating translation factors and ribosomal activity through phosphorylation, these pathways regulate protein synthesis rates in response to environmental cues, ensuring efficient adaptation to varying cellular demands and external conditions. Thank you for watching, and don't forget to subscribe, like, and share.